Heredity. In this capsule, you are going to study about monohybrid cross experiment. Let us recall a few points of the previous capsule. Here is a quiz for you. Who is called the father of genetics? Answer: Gregor Johann Mendel. Name the plant selected by Mendel for his experiments. Answer: pea plant or Pisum sativum. Name any two contrasting characters in pea plants that Mendel selected for his experiment. Answer: 1. The height of the plant, tall and dwarf. 2. The color of the flower, that is red and white. Now let's revise a little bit again about pollination. What is pollination? It is a process of transfer of pollen grains from the anther of a stamen to the stigma of the pistil. Now list the agents of pollination. They are wind, water, insects, mammals, birds, etc. Pollination occurs in two ways. One is self-pollination and the other is cross-pollination. What is self-pollination? It is the transfer of pollen grains from the anther of the stamen of a flower to the stigma of the pistil of the same flower or a different flower in the same plant. So what is cross-pollination? So transfer of pollen grains from the anther of the stamen of a flower of one plant to the stigma of a flower of another similar plant. What is fertilization? It is the process of fusion of male gametes in the stamen with the female gametes of the pistil of a flower to produce fertilized egg or zygote. Now let us study Mendel's monohybrid cross experiments. What do you mean by monohybrid cross? A cross between two pea plants which differ in one specific character is called a monohybrid cross. For example, color of the flower in a pea plant, that is red and white. It is one contrasting character, hence monohybrid cross. But Mendel, for his experiment, took another contrasting character. That character is height of the plant, namely tall and dwarf plants. To obtain this single character in pea plants, he self-pollinated the pea plants for several generations to obtain pure tall plants. The similar procedure is what he tried for the pure dwarf plants also. Every time when these seeds were sowed, tall seed plants would give tall pea plants only, and dwarf seed plants would give dwarf pea plants only. Let us see another example performed by Mendel. Mendel cross-pollinated a tall pea plant with a dwarf pea plant. He repeated the same experiment, taking a pea plant with red flowers and another pea plant with white flowers. First, let us understand a bit about one characteristic of the plant, that is, height of the plant. For better understanding of Mendel's experiments, we have to know something about chromosomal nature in a cell. Every somatic cell contains two sets of chromosomes, one from the paternal side or the father and another from the maternal side or the mother. Here in this chromosome from the paternal side, there is a gene responsible for height or tallness. And the chromosome from maternal side has another height gene responsible but for the dwarfness. He transferred the pollen grains from the anthers of the stamen of the tall pea plant to the stigma of the pistil of the dwarf pea plant, or vice versa. He cuts the pollen grains and covered it to avoid further pollination. After some days, the flower withers and falls off, and the ovary forms seeds or fruits. He collected the seeds and what did he do with them? He sowed them in the next season. He expected the hybrid or the crossed plants to be intermediate in their height. But to his great surprise, he found all the plants to be tall. He called these hybrids the first filial generation or F1 generation. Here, he self-pollinated the flowers of these plants. He then collected the seeds of F1 generation's plants. Now let us see the chromosomal changes that happened from the parents to the F1 generation. In the parents, after meiosis, sex cells are formed. Here, let us concentrate only on the height of the plant. The pure tall tea plant is called capital T and capital T. Do you know why two capital T's? Because I already told you that every somatic cell contains two sets of chromosomes, one from the father and one from the mother. That is one capital T from the father, another capital T from the mother. So next, let's look at the pure dwarf plant. What may be the pure dwarf pea plant? It is small t and small t. Now you may get a small doubt. Why small t? Why small t? Well, you will know the answer after this experiment. But tell me again, now that you know some part of it, why two small t's? Yes, one from father and another from the mother. Very good. After sex cells formation, capital T is a male sex cell and small t is a female sex cell. During pollination, these combine in this particular manner. For better understanding of the same concept, we use the punnet or checkerboard. After pollination, the seeds had factors, capital T's and small t's. In this generation, all parents were tall, that is one capital T and one small t, and he had called this F1 generation, that is the first filial generation. In the next generation, he planted the F1 generation seeds. He found a mixture of tall and dwarf plants in the ratio 3:1. He called this the second filial generation or F2 generation. Let's see how self-pollination takes place using the Punnett board. In F1 plants, capital T small t, the gametes are capital T and small t. Finally, the result. The F2 generation plants had 3 tall plants and 1 dwarf plant. Hence the ratio 3:1. Here were the 2 tall plants where one was with 2 capital T's, other 2 were with 1 capital T and 1 small t. Can you tell me why both capital T's and another capital T and small t became tall? This is because of one law of genetics, which means that if one dominant characteristic is present in a gene, it will suppress the characteristic of another gene. It means that in this experiment, capital T meant for dominant character expresses tallness. Small t is meant for recessive character. Hence, capital T both dominant character shows tallness. One capital T suppresses the dwarf character, that is small t. By this time you would have guessed why small t and small t is a dwarf plant. Here there is no dominant gene to suppress dwarfness. Mendel repeated the same experiment with other contrasting forms of characters. He obtained the same results in the same ratio of 3:1 in the F2 generation. This ratio is called monohybrid ratio. For example, if you take the color of pea seeds, one is green, that is capital G, and the other is yellow, that is small g. After F2, what might be the color of the pea seeds? 3 green and 1 yellow seeded plant, that means 3:1. Now, can you guess the dominant character? That's right, the dominant character is green color of the seeds and the recessive character is yellow color of the seeds. Now the next contrasting character, round and wrinkled pea seeds. Now tell me, which is the dominant character and which is the recessive character? Round shape of seeds is the dominant character and wrinkled shape is the recessive character. Here the contrasting character is the color of the flower of the pea plants. Now tell me, what would be the color of the flower here? Yes, you're right, because it is going to be white because that is a recessive character here. Red color of the flower is a dominant character. So here also 3:1 result is obtained. Mendel then began to analyze and interpret the similar results obtained in all these experiments. He presumed that each contrasting form of character is controlled by a factor. That is, there should be a factor for tallness, a factor for dwarfness, a factor for producing yellow seeds, a factor for producing green seeds, and so on. In the experiments where the tall and dwarf were crossed, the F1 plants, though all tall, must have contained the factors for both tallness and dwarfness. This is because when F1 plants were self-pollinated, the F2 generation produced some dwarf plants along with tall plants. The factor for dwarfness, however, remained hidden or concealed in F1 in the presence of the factor for tallness. Mendel called this factor for tallness dominant over the factor for dwarfness, which he called recessive. From this monohybrid cross experiment, we can understand that the F1 plant had therefore inherited a factor for tallness from its tall parent and a factor for dwarfness from its dwarf parent. Mendel then finally arrived at a conclusion. He stated, when two factors responsible for a pair of contrasting forms of a single character come together, normally one will express itself while the other does not. For any given character, there are two forms of factors. One factor is dominant and the other is recessive. This idea is known as the principle of dominance. Now it's time to think and answer. What is meant by monohybrid cross experiment? The answer: a cross between two pea plants which differ in one specific character is called monohybrid cross experiment. What is the F2 ratio in monohybrid cross experiment? Your answer, it's 3:1. Discuss with your friends. Name the type of pollination used to obtain F1 and F2 generations in monohybrid cross. Next question. Ramya conducted monohybrid cross experiment considering the color of the pod of the pea. In the F2 generation, she obtained 3 yellow and 1 green pod. Discuss this with your friends and make a report. Please copy this down. In this capsule, under monohybrid cross experiment, you have studied about F1 generation and F2 generation. That is, when pure breeds were cross-pollinated, we obtained all tall plants in F1 generation. And when F1 plants were self-pollinated, we obtained 3 tall plants and 1 dwarf plant in F2 generation. So the ratio is 3:1. In this capsule, the key words are hybridization, self-pollination, cross-pollination, monohybrid cross, pure breed, filial generation, monohybrid ratio, that is 3:1, dominant, recessive, principle of dominance, Punnett square, and F1 and F2 generation. Here are some activities for you to do on your own. In your school garden, grow pea plants using different colored seeds, pods, etc. and observe their growth. Next question. In the above activity, try to self and cross-pollinate plants and make a report on your experiments. Thank you.